All right, good morning, everybody. Thank you for tuning in. My name is Jill Goodwin. I am the conference manager for iDig Bio. And today we have Ed Stanley, who's going to be talking about lessons learned from oVert, elucidate a path forward for fair collections networks. And Dave Blackburn may or may not be tuning in a little bit later. But for now, we have Ed. And thank you so much, Ed, for being here. And I will let you take it away. Thank you so much.
Okay, thanks, Jill. Thanks, Kit. Good to see you guys. I can't see anyone else out there, but hello to everyone out there. So I'm going to talk a little bit about some transitional stuff that we're doing from our big TCN that focused on digitizing museum specimens to the next stages and the stages after that. So I wanted to start, for those who haven't heard about oVert before, by talking a little bit about the plans for digitizing museum collections. So our museum holdings, our specimens, are a lot of things, right? We have our samples, our specimens that we've got out in the field and preserved, and we have the physical objects. But then there's a lot of additional data around those physical objects, right? Things like sound recordings and information about the collection event itself, DNA, tissues from the sample that are stored elsewhere or separately from the physical object. It's all part of that extended specimen. And accessing that data is challenging depending on what kind of data we're talking about, right? Some of that data is very easily shared. So things like sound recordings, even the old, reel-to-reel tape recordings, can be digitized and stored online in places like the Macaulay Sound Library, which principally does bird songs, but it also has a lot of frog calls and other things. If you're watching this video, this recording, you probably know about iDig Bio. So places like iDig Bio are great for data aggregation of digitized specimen holdings. So it kind of pulls all of the information about collections, collectors, dates, times, that locations, that sort of thing, pulls that into a single centralized place. And then, while the tissues themselves are quite hard to, are physical objects, and you can't digitize them and share them. The tissues themselves, the DNA and the DNA sequences that are extracted from that are just A's, T's, G's, and C's, right? It's text, so you can store that and share that through websites like GenBank. But the physical objects themselves, the specimens, where I think a lot of people think the kind of the majority of the data or information about the actual physical objects is harder to share, right? Harder to share over online. You can't email a frog to someone. And so the samples tend to sit on a shelf or in a folio or in a box in a warehouse somewhere and wait for someone to come and study them, or if the need is sufficient and the requester is trustworthy enough, get a loan out to have it sent. Right? So they're sitting there waiting to be used, and how they're used is another stumbling block for access, right? Because if you want to use destructive sampling, if you want to take the object, the specimen, the sample, and change it in some way, you have to have a good reason for doing that. The rarer and the more important and the more destructive the sampling you want to do, the better the question needs to be in order to sacrifice that object, right? So if you only have one specimen of a species, and that's the only thing that was ever caught, and you want to skeletonize it, you have to have a very good reason for doing that. So non-destructive digital sampling is something that lowers the kind of the activation energy for a lot of these loans, and of course, if you can digitize it and email it, that lowers the energy even more, because it doesn't cost as much money to email or download something as it does to post something in the mail, and it reduces the concerns for being lost and things. So physical digital imaging, like 2D digital imaging, like photographs and 2D X-rays, is something that people have been doing for a while now, but it's limited, right? Of limited use. You need specific photographs and specific orientations, and if you have particular questions. So it's not a kind of a one-size-fits-all type solution, but recent advances or access to things like CT scanning, micro CT scanning, allows us to essentially take these objects and look inside them digitally in 3D without damaging the sample. So, a CT scan is essentially just a 3D X-ray. So we can take our sample, and we can CT scan it, and we can take that object and look at in high resolution of things like the skull, and we can apply color gradients to see where the high and low density areas are inside the skull. We can crop through things, digitally. We can apply different color layers to it, so we can see where the thick and the thin areas of the skull are. We can even look at negative space, so endocast, so we can kind of take the space inside the inner ear and visualize that in 3D. And that's all great for the skeletal information. But we can also look at the soft tissue by soaking or treating these samples with contrasting agent, and that allows us to see the soft tissue. So we can peel off the skin and look at the muscles and the blood vessels and the nervous system, the digestive system, and all these other parts independently. So we can isolate these things out, we can segment these things out, but then put them back together again, right? So this allows for multiple dissections of the same object, and at the end of the day, that sample goes back on the shelf, and it's ready to be used by other people. And so CT scanning is one of the most commonly used way of digitizing museum samples, but because it's an X-ray based approach, it doesn't capture real color, it's density only. So things like photogrammetry and light scanning do allow us to visualize the actual true life, or at least the true specimen colors of these objects. While there's no internal structures recorded through these things, the external anatomy, if you want to look at the kind of the morphology of the feet or something on this toad, we can do that too. And so in addition to getting lots of information out of these samples, we also have a way of sharing this information on databases like Morphosource now. So that's another additional way of getting that information, that data, that specimen information out there. And so this isn't the first. Our project wasn't the first time that people were doing this, right? This is technology that's been around for a while now. And so people have been looking at these data sets and building these data sets and putting them up online and sharing them, but it's been in a somewhat piecemeal way, right? Because it's expensive and it takes a lot of time and energy and expertise to digitize these kinds of objects. People tend to focus on specific research questions. So sticklebacks, you might scan a handful of different species of stickleback, or you might scan an entire big group of lizards like anoles to understand morphological variation inside this group, but that's essentially circumscribed to particular questions. And of course the tree of life is enormous and very diverse, and if you only sample the things that people are actively working on, you end up with a representation that looks very piecemeal and not particularly good for building large-scale evolutionary-based hypotheses. So we were hoping, with our plan back in 2017, was to essentially create a scaffold, or at least not a comprehensively sampled, but a broadly sampled scaffold of CT data across the vertebrate tree of life that we could use to supercharge a lot of these new evolutionary biological studies using CT. And so that was the openVertebrate project, or oVert project. And it had about 50 different institutions involved in it, loaning samples and scanning data and storing data and processing these data sets. Our idea was to scan one of every vertebrate genus in US collections. The genera spanned the world, but these were holdings of US collections. We also were trying to get one of every vertebrate family contrast enhanced scans, so that's with the soft tissue visualized. All of that data went up on Morphosource, which is a free to download repository of museum-based data. And then for a subsection of that, we further processed these data to make them more accessible to educators and the general public. And so it was was it a 6-year project, I think. In total, we were funded for 3 years, I think, and then had a couple of no-cost extensions because it was just such a big job. And we did a lot of damage to the Morphosource holdings there. We put a lot of stuff up online. So nearly 30,000 media, nearly 15,000 specimens. That's been downloaded nearly 150,000 times by 3,700 users from a bunch of different countries around the world. The sampling we tried to get as broad as possible. Things like birds were a little bit of a challenge because it turns out that it's harder to find alcohol-preserved birds than it is to find skins and skeletonized birds, and we were predominantly focusing on the wet collections. But sharks, actinopterygian fish, amphibians, reptiles, and mammals, more or less, we did pretty well. And so Morphosource is the site that we partnered up with. Well, Doug and Julie are part of the grant, but this is the site that we put it on, and it's a pretty I think intuitive and powerful website for people to access this CT data. So in addition to having kind of a preview function and all of the information associated with the specimens there, you can visualize the slices now and see see what you're about to download. So there's a lower downsample resolution of these volumes, but you can also visualize the volumetric renderings as well to see to get a better idea about what the media you're about to see is. And this has been a big, this has been a big success. We've had a good number of papers come out of it from the oVert group, but also people who have not associated with us at all and just used our data. We've built a number of educational applications for this, including some cool reimaginings of those old chestnuts like homologous structures in the forelimbs of vertebrates. We hosted a lot of high school teachers to come in and learn how to use these data. And it's been the data has been used in all kinds of range of different outreach things like museum exhibits and art exhibits and things like that. So this the final project finished up last year, and we had a lot of I think positive press that came along with the publication of the oVert paper. But we also, I think, learn some things that'll be useful for new TCNs and new people trying to do a similar sort of thing. Essentially, we ran into some impediments to use of this data, and that's partly because of the data itself, and partly because of the structure of places like Morphosource. But ultimately, this is what people see when they find their samples on Morphosource, right? It's a cross-sectional image. There's information about the media type, and there's some language about what was what this sample was, how the sample was made. But ultimately, I think people want this, right? They want the end product. They want the thing that they can immediately pull up and ask questions of. But to get to this kind of image, you need to download these tomograms, which are somewhat abstract looking cross-sectional images, recompile them into a volume, and then segment that out with some specialist software. And there's some free software out there, and it's very powerful, and it's pretty intuitive, but it still requires some kind of activation information to get there, right? And so one of the things, we got a lot of positive feedback for when the paper came out, but then I got an awful lot of emails saying, "This is amazing. I love this. I don't know what I'm looking at. I don't know how to use this." And so the idea of accessibility not being the same thing as availability, right? Just because someone has access to something, it doesn't mean that that data is available to them, right? Just because it's available doesn't mean that they can access the information within it. So those two things, I think, gave us pause. And you think about it like an amazing library that has every book in the world in it. But if all of those books are in a language that you can't read, it's not of use to you, right? You can walk in the library and take books off the shelf, but you can't access the information within. And so it's kind of frustrating for folks to run into that. And so that's a methodological barrier. But the another barrier is kind of a slightly simpler one, more straightforward one. So Morphosource is a website that was designed by museum scientists for museum scientists principally. But if we're wanting this data to be used by as broad a number of people as possible, there's some knowledge gaps that prevent people from getting access. For example, if you go into the search term and type in chicken, you'll find nine media, all of which are embryos and eggs, which is very cool. Awesome, awesome that that's freely available to people. But if you type in Gallus, the genus of chickens, you find a lot more, including some fairly useful material that people could use without having to do a lot of processing, right? So the prep samples are there, but the access to them is not as clear because people don't always know the Latin names of these things. So we made a video. Our postdoc Jamie Gray made a lovely video explaining how to do that and walking people through that. But it's just an additional step. One of the things I think I like most about the openVertebrate project, and that I think my understanding of the system grew more, was building this pipeline, right? Understanding how you go from creating data to storing data and accessioning data and making it available, archiving data essentially. And so there's a lot of moving parts. We had a lot of different museums and scanning facilities and other places that were involved in this, and so building a pipeline that standardized things and made things available the same way to people was challenging. And one of the things that we realized as we went on, or I realized, I suspect other people knew this before, there's an awful lot of databases out there that have like important, unique information inside them that don't necessarily, you don't need to reinvent them, right? So IDIG-Bio, of course, aggregates all the data from Specify and other places to put it into a single stored place. And so that's all specimen collection data. Morphosource is 3D and 2D morphological data sets. Things like the RRID site have research facility information. So that's the labs and the machines inside those labs. ORCID is sure we all know, ORCID is essentially a way of storing information about the scientist, the person, the individual. Futures compiles trait data. GenBank has genetic information, genetic data, sequence data. Uberon is an anatomical ontology atlas, so that's a lot of information about how all of that fits together. And then you have things like the Macaulay Sound Library and other places that have sound data, right? So there's lots of little parts that all tie together. And the interesting, or I think the kind of the exciting part about how this all works is that you can build a network of networks and connect them. So you don't necessarily need to reinvent. You don't need to gather up the information about the collecting event de novo. You can just pull that directly from Morphosource. You don't need to necessarily identify all of the structures in the arm. You can pull that in from of a CT scan that you've done. You can pull that in from Uberon, and you can link sequence data back to the morphology and then ORCID. You can ping people who've done particular things and list them. So it really reduces a lot of the work that's required of compiling these kind of databases and these archives. So that's really exciting, and I think that's going to hopefully come back in a little bit in the future. So the other thing we realized as we were doing this is that as museum scientists, we've done this for a long time now. We work with information-rich data sets, right? Not just CT data, but the physical objects themselves, right? They're incredibly, almost infinitely rich sources of biological information. And we apply a diverse range of uses and methodologies to those samples, to those specimens. And I think this is the most critical part, we have an intrinsic mandate to store these things and make them available for people. and maintain them for people long term. And so that really speaks to a big push from NSF that's focusing on the FAIR principles, or findable, accessible, interoperable, and reusable principles. So the office, the White House Office of Science and Technology put out a mandate, or a memorandum a couple of years ago called the Nelson Memorandum, that basically outlines that if you're using funds from the government, if you've got a government federal grant, you have to make your data available to the people who financed it, which is the taxpayers. And so focusing on these FAIR principles is going to become more and more important. And museum people kind of that's our bread and butter, right? That's part of our ethos in the first place. So back in 2019, no, 2020, we started talking amongst ourselves about potentially putting in for one of these FAIROS, findable, accessible, interoperable, reusable, open science research coordination networks, and a group of us got together and wrote a grant to basically take what we'd learned from our experiences with oVert and other places, and apply that broad scale to tomography data in general. And so these are the people, and it's worth noting that most of these people, in fact, all of the PIs and co-PIs, at least either work at a museum or work at a core facility that serves multiple user groups. And so, the idea of sharing data, making data available, archiving things is important. And so NOCTURN, the Non-Clinical Tomography User Research Network Group, really does focus on applying FAIR principles to tomographic data. And that sounds like a big project, and so conceptually conceptually or methodologically circumscribed, but conceptually quite large, right? So lots of people scan lots of things for lots of different reasons. We obviously focus on things like got chameleons and lice here, but biological museum specimens, but people scan geological samples, electronics, light bulbs, whatever. There's a lot. I think this is an injection cast wind turbine blade, right? And that often those things get scanned as well, because lots of important internal information on these things. So so a lot of people scanning a lot of different things, but ultimately, although these objects are all very different, their fundamental data associated with the scans are just two-dimensional cross-sectional images with a gray value. So the darker the pixels, the lower density of the object that pixel represents, and the brighter it is, the higher density it is, right? And that's the same for whether it's a light bulb, a chameleon, or an electronic chip. The challenge, of course, is that there's tons and tons of different types of machines and different ways of scanning things. So the CT algorithms and the CT data, the approach themselves is fairly consistent, especially with micro CT. It's just a revolving series of revolving images of X-rays, but the manufacturers and the software that these data are produced by are wildly different, and so there's a lot of disconnect with the vocabularies and terminologies. So the first thing that really the NOCTURN group were really trying to do was try to tease all that apart. So the structure of NOCTURN is essentially divided into 4 different working groups that correspond to the FAIR principles, so findability, accessibility, interoperability, and reuse. And then that's binned into 3 different communities. So we purposely sought out members of the educational community, museum people, artists, educators, teachers, that sort of thing. Basic and applied research. So that's both the kind of academic basic research, but also applied research groups as well. And then industry and vendors. We thought that was important because there's so much disconnect between the various different machines and softwares that it's important to bring those folks in. The working groups and the community groups meet once a month online. We have one virtual meeting once a year. And the idea is that, there's enough cross-pollination between the groups, right? People attend multiple different communities and working groups, and so there's enough interdigitation, interweaving of those groups that people will have an idea about what's going on. But once a year, we have a digital meeting where we all touch base and make sure we're not duplicating effort. And then in 6 months after the virtual meeting, we have an in-person meeting. So we're going into our 3rd year now, and working on that. So just really briefly talking about what those different groups are focusing on, because I think this is probably quite useful. A lot of these things were guided from our understanding of how people access data and find data from oVert and through Multisource, but there's a lot of non-museum folks involved in this as well. So it's kind of an interesting to see other groups' ideas about these things. So the findability group really working on things like persistent identifiers, again, something that museums are pretty good at, right? We have our Darwin core triplets and, and our identifiers that tie specific samples to particular data sets and metadata. Working on repository standards, what are the terms that people want to be recording and storing and searching. We've built a global map of CT labs, especially CT labs that are open to external users. And then focusing on a series of best practices of a good, better, best for storing and archiving data. So what kind of data do we need to make immediately findable, right? What's the data that most people are going to be using, and what data do we need to store, but we don't need to prioritize its findability? Accessibility working groups, an interesting one we're working on because there's lots of different types of file format outputs. We're working to try and find, if there's not a monolithic solution to this, at least what are the unified characteristics of the successful ones. So, not necessarily you don't have to use this format or this format, but if you're going to use a format, it's important to apply, that this format agrees with these things. Understanding how metadata persistency works, and building essentially a database of scanning events. And so we recognize that, talk about this in the reuse section, but it's hard to build, it's expensive, it's very expensive to build these large archival sites, right? The data from CT is quite large, and so that requires a lot of storage, and scalability is a real thing with this. But a record of what's been scanned and who has that data, and who is responsible for that data, and who is safeguarding that data, is a lot cheaper, and it's a lot lower risk. And so it's a bit of a stopgap between the obvious perfect solution, which is everything that's scanned goes up onto a freely accessible and infinitely scalable database. But until that happens, having some kind of record and kind of a phone directory almost for or an email directory for how to get hold of that data is another thing. We've written scripts to automate an extraction of metadata from a range of different machines, so you can kind of pull the same data out from different kinds of different types of metadata files. And then, because again, accessibility doesn't necessarily mean availability, we've been working with the outreach and education community to really develop educational tools to what to do with the data, what to do with the raw data, the tomogram data, and how to get access to that. Our interoperability working group is a really interesting one. So this is the one that principally is tackling the mismatch in terminology and vocabulary between different machines. So we've kind of built a sort of a Rosetta Stone, right? If your GE system puts out this value, that actually corresponds to this value, or this term in your X-radius system, and this term in your North Star system, right? So it's a way of linking. All the machines are essentially doing the same thing. They just call the components different things. So this is a way of linking through that. The interoperability group are also exploring ways of reducing the bottlenecks for image processing. right? So you know, we're lowering the cost, but also the time involved in processing data. And then and this is a little bit specific to CT data, but essentially your gray values that represent density don't necessarily represent the same density across different data sets. So if you scan a big rock and you scan a small I don't know, insect, a ladybird, you know, they will have the same range of densities, but of course the density of the rock will be much, you know, the gray value associated with the rock will be much denser than the same gray value associated with the ladybird. So introducing these things called phantoms, which are objects of known density that can be scanned alongside the data, the object, you can use that to calibrate and actually make your data a lot more interoperable, right? So the gray values of data set A actually translate to the same density as the data set B. And then finally, our reuse group is working mostly with the repositories, working on sort of boilerplate data usage licenses, or at least how to phrase data usage licenses depending on the kind of things that are important to you. Working on data and metadata provenance, so tracing you know how tracing who uses the data and then the downstream derivatives of that data. Talking about how the minimal attributions, the minimal information that you need to report, at least if you're going to report anything, these are the things that you have to report in publications, and tying that back to DOIs and linking that back to the data sets. And then this is something that's really come up in the last, since we started the grant, is the idea of getting all of our data at least ready for these large scale neural network or machine learning AI processes. So, we put most of our information out through either through the website or through publications. So our website's, you know, probably the most dynamic of those things, right? So that we have our CT Rosetta Stone vocab list up on the NOCTURN site. That's nocturnnetwork.org, one N in the middle of that, because not the N of Nocturne is network, so I don't like tautology or anything with these acronyms. So yeah, so nocturnnetwork.org. Our CT vocabulary is there. We have a career forum, so you know, if there's any new jobs being posted on digital imaging, that will be listed there. That's where our interactive CT map of the world is. If you go on there and you have a CT lab that does do external scanning and you don't see it there, there's a lovely link you can click to register if you want to be on that map. We have our data management and sharing plan templates and boilerplates for verbiage of management plans. And then you know, some op-eds written by people on how to access data sets in online repositories, especially Morphosource in this case. That's really what created that how to access Morphosource video was through the NOCTURN group. So we have a lot of we publish several papers, including, we were mentioned in the NOCTURN group was mentioned in the openVertebrate paper that came out earlier this year, rather. But we have had some peer-reviewed papers on things like how to build a network when nobody within the network understands what the other group doing in terms of, you know, how to build a network with engineers and biologists and educators and industry folks. And our when we were building the world map, we were really struck by just what a kind of a broad range and geographically broad footprint CT has these days. Micro CT and nano CT has, and so, you know, sure, there's a lot of labs in the places you kind of maybe expect them to be, you know, in the US and Europe, but there's a growing number of laboratories that do a lot of micro CT in other spots of the world as well, lots in South America, India, South Africa, Australia, China, Japan. So we're in the final year of the NOCTURN grant. And that doesn't necessarily mean that we're saying, you know, we've mission accomplished, you know, and we can go home. There's lots more to do, and in fact, you know, as we solve smaller problems, the larger problems become more and more apparent. So one of the things that we're interested in is the idea that, you know, digital data is a lot more agile than you know, the physical objects that these data represent, and so it can cross international lines, and the, you know, the opportunities for international collaboration have greatly improved from this. And access to different parts of the world's resources, whether that's museum samples or technology or other things, that's all greatly empowering for people in all kinds of places, right? So as an American scientist, I'm a British scientist living in America, you know, having access to the collections in the Indian museums through collaborations with folks there and CT data sharing that, that's very powerful for us. And it's also very powerful for them because that allows them to kind of get access to our data and collaborate. And so the next real steps are taking this the network that we've built in North America, NOCTURN, but then reaching out to some folks in Europe, specifically the National X-ray Tomography Group, Computer Tomography Group, NXCT in the UK, but also through the Tomography for Scientific Advancement group that is initially was a British and a European collaboration, but then has a North American chapter that also works very closely with NOCTURN to build a kind of a longer lasting enterprise that will keep the momentum going for a lot of this work. So just to finish up, really, we're the next few months we're putting in a couple of grants, or at least one grant, to focus on an international collaboration that will focus on global standards, universal scan records, building up international repositories, right? And that can be stored in one particular country or many, decentralized, but as long as there's a kind of a front end that is consistent across that, that's fine. Preparing all our data to be as AI ready and as interoperable as it can be. And then working on open access approaches to 3D data of all kinds. And that's going to be through a couple of NSF grants on our side and some European grants over on the NXCT side. And so that's really all I had for you today. That's kind of the update of where we're at and the kind of future direction. So I'd be very happy to chat with you more about where this can go and how this will manifest itself. So thank you all for listening.
Thanks, Ed. That was great. For folks who are listening, feel free to put a question in the chat. Greg Watkin-Cowell asked, how long was the NOCTURN grant for?
It's just a 3-year grant. And so, and it's a research coordination network grant. So it really wasn't, again, just like the TCN, it wasn't really to do any research, and it wasn't any, these RCNs tend to be getting a lot of people together to talk about the problems and working out, you know, how to solve the problems, but not necessarily writing the, you know. So we wrote some scripts and we built some things to solve the problems because we were frustrated about, you know, the lack of availability of those things. But that wasn't in the mandate of the grant. It's really a research network grant. So the next group will be a lot more targeted, be able to hire people to, you know, to build code, to write code, to build more infrastructure, not necessarily the kind of the repository infrastructure, but, you know, to actually be able to produce more manifested solutions to this. Yeah, through 3 grants, 3 years, and we had money for 3 in-person meetings every year, and then we made it stretch a little bit further by doing a lot of online things. Thank you, Greg.
Also, this is a webinar, but if anyone would like to just raise their hand and ask a question and join in discussion, that you're welcome to do that too. And Dave, I see that you're here. Dave Blackburn, did you have anything that you additionally wanted to add? I don't mean to.
Well, now I will.
But,
yeah, I'll just say, you know, as Ed knows, I've often was asked, what was oVert part 2? Is there another grant? You're going to scan more things? And I really came to think as NOCTURN as oVert part 2, that, it's easy to make things available, but it's a lot harder to make them accessible and a lot harder to build the community of people around those data. And, IDIG Bio has done that well through lots of webinars and trainings, but I think a lot of the TCNs were kind of designed as data generators and maybe not as much of thinking about like how you train the community to use them. And I do see those things as kind of going hand in hand. It's nice if they're funded in the same project, but I think sometimes, we can use these other funding mechanisms at NSF to sort of stagger them. One project is generating data, the next project kind of builds on that as more training and education and building connections. And actually kind of like the idea of thinking about those different funding mechanisms as sort of staggered and scaffolded on each other, as a way of kind of promoting the use of a lot of biodiversity data, throughout the community. So.
And the kind of the path isn't always a straight line, right? So funding a 10-year project, if this was funded as a single project, NOCTURN and oVert, it would be an entirely different beast at the end of that than it would have been, as we split it up. So it allows you to kind of react to the kind of the limitations, the kind of the impediments that you're running into, and identify where the kind of the triaging needs to happen, and be a little bit more transparent about that.
I think the other thing I'd like to add is that the oVert, and probably true for many TCNs, a lot of the attention is paid specifically to the data generated. I think the long-lasting impact is less that. I think the long-lasting impact is sort of changing some perceptions about collection's role in generating and managing data. I think, there's countless projects, basically sort of in the oVert mold, that are either just individual research projects where now the ethos is, this is what we do with our data. This is how museums interact with our data. I mean, they're sort of stepping into the shoes of oVert on a smaller scale. I think that's a pretty cool impact, and I think really that all comes down to the people side of the TCNs, and that's sort of what NOCTURN picked up, was just building out that network of individuals, and that sort of ethos in the community. And you see, as just Ed said, it's not linear. Some of the kind of impacts long term are, they might seem sort of outside the scope of the original project, but like one interesting one I've always found is that over the last, let's say 5 to 6 years, the number of CT scanning grants, like grants to purchase a CT scanner that have gone into the National Science Foundation's Major Research Instrumentation Program, has really increased. And not only has it increased, but it tends to be increased at museums and universities associated with museums. And so that's kind of neat, that there's people interested and willing to kind of invest in the technology to kind of support effectively becoming a node in a network that no longer really exists, but that's fine because they're sort of a social network, officially. And I think those are kind of the long-lasting impacts. Hopefully the data are useful for a long time as well, but I think more of that sort of community network and mindset about how we, what is our responsibility as an institution when it comes to data, sharing data, building resources, making connections. And it's neat to kind of see, and I hope something that other TCNs or similar types of projects will pick up is that, there's the making it available, but then there's all this other work that kind of happens to really make the whole thing sing in the end. And there's some interesting extra funding opportunities out there, just like the RCNs. So that's it.
Yeah, I mean, I'm just jumping in because I'm afraid of silence. But also, it's an important point that, that tree of life thing I mentioned before. The idea was that we weren't going to scan every single thing, right? We just wanted to create a kind of a framework with the and as Dave mentioned, the kind of I think one of the real strengths of this is is building up the awareness and the impact of what this kind of data can do, and then providing a series of clear and easy pipelines to pay back into that data set and into that repository as well. So streamlining some of the uploading processes, linking the linkage in between iDigBio and Morphosource is one of my favorite outcomes of that. To building those connections that like take some of the weight off the user to add that in, and then building additional networks, building in additional, ORCID, RRID, those kind of things. That's all part of that, right? And so the idea is that, yes, it's nice to be able to, do a pan-avian analysis of a particular thing, but when you fill in the gaps that you need to fill in, that goes back in and that makes the database even stronger. That makes the repository and the archiving even stronger and more useful.
So, go ahead.
Yeah. No, we just have a question in the Q&A, and is there any chance for more training workshops at conferences on things like how to use a TIFF stack?
Yes, we do actually. So one of the things that I didn't mention in the NOCTURN thing, but we do have a pretty good presence across some of what I would consider to be kind of the classic meetings like SICB and the ICBM and things. So we have, in our little part of the world, we certainly do have we have presence there. I think it'd be great to have more workshops at these meetings, especially when there's more open source softwares that really kind of reduce the cost of buying for this thing. So yes, I think that is part of our plan. And if you have a meeting that you'd like us to attend, I think that would be fun and fine to do.
Also, I just have this is a very strange question. So if there's not an answer to it, that's fine. So I've mainly been working with NetCDF file formats. And there's a lot of metadata that's available. And that's typically what's used for NASA geospatial, and NEON uses H5, which is basically NetCDF4. Has there been a lot of talk about interoperability between file formats in museums, CT scans, these really large files, and these more geospatial ecology files? So maybe it'd be easier to work with some of the CT scan specimens in context of their geographic region. That's a weird question. It's okay if there's no answer to that.
Dave, do you want? I mean, I have it.
Yeah, I think there's a two-part question, really. One is that, and probably what Ed would say is that, there has been a lot of discussion about sort of simplifying file formats so that some of the work that's done with CT scans. So there's the image stack, and then there's bringing it into a piece of software, which could be proprietary, it could be open source, and then doing all the manipulations. And so one challenge is that some of those file formats do not port between different types of software. So I think NOCTURN has opened the door to having those conversations. So they are happening about having, can there be some more universal type files for dealing with volume data, for dealing with the segmentations of the CT scans. But you also asked about this interoperability of basically phenotype and space. So one thing that oVert didn't do it. We paid for it before oVert started, and I think is sort of one of my proudest accomplishments for it. The iDigBio API has not been used extensively by a lot of other data aggregators. And so one of the things that we built into Morphosource, actually, and I paid for this before Morphosource, the oVert project began, was to basically have iDigBio have the Morphosource use the iDigBio API for bringing in metadata from specimens into Morphosource, so that we could make more clear linkages of the metadata that come out of a museum in their record set, end up in a data aggregator, and then end up being the same type of data that ends up, in Morphosource. It doesn't have to be all the data, but because we can link those, it means that it would be easier to crosswalk back to say, like, I have skulls, like, can I find coordinates for them? You know, if you try to put genetic data on a map using GenBank, good luck, right? There's very little connections between the original collections data and the sort of derivative data of sequence data. But using the IDIGBio API was a way that, I mean, you can't do it quickly, but it is possible to basically crosswalk back to the collections data and get points on maps if you want to, you know, say like, yeah, I want to look at geographic distribution of skulls on a map, right? Like you can do that because we kind of built in these linkages. So it is ultimately findable. Yeah, I'm going to have to head out here in a second, but.
Yeah, and I'm just going to follow up on that, and then that's entirely based on those UUIDs, right? The universal unique identifiers, and being able to create those linkages between these things. So having, you know, and that's why these persistent unique identifiers are critical, right? Because you need to be able to link your morphosource data to a specimen, and that specimen can be linked to, you know, a locality, and that locality can be linked to a map layer that tells you the temperature of you know, average annual temperature or whatever. So you could, you know, I mean, in theory, you could write it. So show me all the skulls of birds that live in a temperature range of this and this, right? That's the kind of linkage you can build, right? You don't need to add all of that data by hand into, you know, one great database. As long as you can link those things, that linkage is I think the most exciting part of that. So, and making sure that that's doable is critical. So, you know, again, there are very few equivalent repositories for engineering and electronics and geological samples. There's the Digital Rocks portal that does that a little bit, but there's no real equivalent to Morphosource. So all of our work with Morphosource and IDIGBio has really has created a kind of a baseline for, you know, how to build these things as people go forward. So yeah, and that linkage.
Oh, sorry, I didn't mean to cut you off. I was gonna say, Greg said that Spinach 2025 is in Kansas, so it'd be really cool to have a workshop on CT scan data use. You want to come back to Kansas? What, did you come to Kansas this year? I can't remember.
I wasn't at Kansas this year. The last time I was there was for the SSAR meetings, which is also so I like barbecue as much as the next person. So where in Kansas is it?
It's at KU.
Okay, perfect. All right, I like Lawrence. Yeah,
and all those people. Yeah.
Yeah, perfect. The last time I went there, I flew into Kansas City, but it was Kansas City, Missouri, and I was terrified that I went to the wrong location, and I was now in the entirely the wrong state. And it turns out that is the thing.
Yes, spinach is happening. I think also too, a lot of these linkages and these conversations around how this different data can work together too is something that I'm really interested in featuring at BioDigicon too in September of next year. I think that, you know, a lot of time these conversations are happening within focused communities, and as they evolve and get more complex, when new folks are coming in, they don't understand where to go for this information or how to kind of join the conversation. So I am going to, with that, thank you, Ed, and I would like to just highlight the TCN, the presentations that we have next week.
Do you want to stop sharing? I'm sorry. Shall I stop sharing my screen?
Yes. Did I already? I think I.
Oh,
you got it.
It's on you.
Jill.
So next week we will have a two-part TCN webinar, iDigBees and Dig In TCNs. We'll be talking at 11 again next Thursday, and you can go to the announcement page to access more information about that webinar. You can also go there for recordings for this session, for this webinar and other webinars too. And I will.
There's a link in the chat for the TCN page and also for BioDigicon.
Great. Awesome. And yeah, again, I just made a little plug, but BioDigicon next year, September 2025, will be combining kind of the missions of the digital data conference and also BioDigicon for one large event that will be happening in DC at the National Press Club. And I believe we have confirmation that we will also be working with the Smithsonian for the reception. So a lot of great things going on. We're hoping to work with a lot of federal partners and hopefully can continue some of these conversations about where some of these gaps in communications are and bottlenecks that, you know, are where people are still having questions about how information can come together, and we can kind of move some of those conversations forward. So, but I think this was a really great example, Ed, of how TCNs can evolve and can turn into, you know, new things depending on the needs of the community. So I really appreciate you taking the time to come today. And I know things didn't exactly work out how you were planning, but that was really great. And yeah, I appreciate you being here today. Thank you so much.
Thanks everyone for listening. Thank you.
All right. Thank you all so much for tuning in, and hopefully we'll see you next week.
Thank you, everyone.
Bye.